Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Okay, we'll go ahead and get started. First of all, thank you for coming, for all of you here, and for all of you in the television audience. Today we have Ed Addison from TeraDiscoveries coming to talk to us about using computers to design drugs. And he's been doing a lot of work with Microsoft and with Azure, and they have taken their work that they did in the universities, and they're now making a company out of it and moving it forward. Ed's been 25 years working in this industry. TeraDiscoveries is a serial entrepreneur, it sounds like. And we've worked with him for probably 3 years, I think, since I first met you here at Microsoft, roughly 3 years ago that you started doing some of this stuff.
Not quite, but closely. Maybe 2.
2 years and change? Okay, so my time goes way too slow for me, apparently, because I'm having so much fun. But it's been a very interesting talking with him, and several times that I've met with him before, and we're going to have a chance for you guys to hear what he has to say about rational design.
Okay, thank you very much. I'm going to talk to you today about TeraDiscoveries. We're a small company of 15 people, some full-time, some part-time, some contractors, some employees, typical startup, quasi-virtual. We have a business incubator where we're located on Davis Drive in Research Triangle Park in North Carolina. Before describing what we're doing, let me just take a minute or two to state where we came from. TeraDiscoveries was a venture that wasn't quite planned with a business plan. It evolved a little bit. A colleague of mine by the name of Lawrence Husick and I founded TeraDiscoveries. We've been working together for quite a number of years through various ventures. He is a chemist and an intellectual property attorney, and I'm an engineer turned entrepreneur. Spent most of my career doing business development. And a few years back, I'm a graduate of Virginia Tech, so I'm a Hokie. And a few years back, the dean of engineering at Virginia Tech reached out to me and others who had venture experience, asked us to come in and take a look at some of their technology. They were doing a tech transfer push. And at the time, and this is before Azure and before the cloud was overwhelming, Virginia Tech had a system called System X, which was a 2,200 node cluster with a high-speed network connecting a bunch of Apple G5 servers together. Some of you may have heard that. They won some awards, and they were number 3 or number 7 or supercomputer in the world one year, but they gradually slipped because they didn't go beyond that. When I saw that, I asked the dean what they were doing to commercialize it, and he said nothing. And I didn't really think we were going to sell anything like that, but I suggested that the pharmaceutical industry needed to start doing high-performance computing on an outsourced basis. And I had agreement about that from a major pharmaceutical company whom we did our first engagement with. And what we did is we formed a consulting company. It lasted a year or two, and then along the way, I was making a presentation at Duke University where I was then approached by their tech transfer director and said, I have the perfect piece of software for that big machine you got. Well, this was Inverse Design, which is a computationally intensive drug design tool, which I'm going to talk about today. And so we ported Inverse Design to System X, and it worked at Duke University on a 16-node cluster. On System X, it worked okay, had a few memory problems. But very quickly, the cost was dropping on the cloud, and Amazon was where we went next. We ported to Amazon because the big cluster that we started out with was not really being maintained in a commercial way, and the cost, even though it was an academic price, was higher than the cloud by a factor of 50% or more. And after we were porting to Amazon, we had an internal discussion and decided that we're going to need a lot of computing cycles, not to do our software as a service, but we had a bigger vision, and that's that we wanted to preemptively compute potential drugs for many proteins from the protein data bank as a way of shortening drug discovery. So we identified 3 sources of big cloud computing. That was Google, Amazon, and Microsoft, and the relationship that stuck was Microsoft. I met Todd Needham a couple of years ago, and we began this discussion. And so our stuff runs on Azure, and we aren't using the other clouds at this time. You know, we're focusing. I guess that's a compliment to Microsoft. So what do we do? What we do is we use proprietary software and cloud computing and design drugs. Now that, if you're a chemist, that probably does a little violence to you. We don't really design drugs. We design drug candidates because we don't do the clinical research. But it does it quite well. And we use a method called QM-MM, if you're a scientist, quantum mechanics and molecular modeling. You do a quantum mechanical model of the small molecule or the peptide, and you do a molecular model of the protein, which makes it a lot more accurate than older methods, specifically DOC, which did all molecular modeling, which was a crude and less accurate approach. Even though faster, it didn't produce really good results. Now that's not all there is to it. That's just the binding calculation. The problem is molecular space is 10 to the 65th big, and so you don't want to enumerate QM-MM calculations, which take about 8 hours using P-dynamo on a single node, or you'd be waiting several lifetimes or more for the answer. So what we have done is we've built and designed a heuristic search algorithm through molecular space. We constrain the space by the properties of the protein so that we can search virtual space, including molecules that have never been synthesized before, and only test some of them in order to do a hill. You know what a hill climbing search is, you're Microsoft. So that's essentially what it does. If we describe inverse design, I've already talked about some of this, but we're searching and computing binding energy, but we're doing a lot more. We're using additional filters. We model water, which a lot of the earlier in silico techniques just modeled the molecules and paid no attention to their properties in water. We look for a lock and key fit, in other words, a very good binding score. And we'll optimize that as we search through space. This is all based on a worldwide exclusive license from Duke University and our enhancements to inverse design. It now runs on Azure. We're still testing a few things, but we have modeled several proteins. We've done the calibrations. We're starting production runs this month. We have 1 patent issued on the heuristic search as applied to chemistry, and we have more intellectual property being filed. So our partnership with Microsoft is to use Azure we've done runs with 500 nodes, and we're trying to up that now to about 1,200 nodes. It could go even higher than that, but there is some iteration in terms of the scores where if we do too many, then we might be less efficient with the computing. We have to keep, what we do is we launch, say, 1,200 QM-MM calculations, get the 1,200 scores, and use those scores to determine which 1,200 to do next based upon the heuristic branch and bound hill climbing search. And then we have to do several dozen iterations. So each one is 8 hours, so several dozen, it's a few weeks for a given protein. Not running continuously for a few weeks, but it could run continuously, but we go from long calculations, score update, repeat, and so forth. And so a little bit of technical detail, and this is an illustration of the idea that we choose the best molecule in the database, but we don't calculate the binding score for every molecule in the database. And we're looking for local maxima in the scores. And so you might have a surface. This is a simplified illustration because the dimensionality is really higher than this. But with inverse design, it's an m times n as opposed to enumeration, which is an m to the nth power computational cost, where m is the number of sites and n is the number of groups per site. And so groups per site has to do with how big the molecular space is. A group is, you can make a small molecule by, I guess some of you may be chemists, and a group, if you put an R group on a molecular scaffold, it's a group. And you can put 10, 20, 30 of those on a scaffold that's typical to a protein, and then that defines a rather huge space. And then the M is the number of sites, which could be, you could think of it in simple terms as a number of proteins, but each protein has multiple binding pockets, so it's really the number of binding pockets. So just to compare inverse design with prior methods, you'll often hear old school biochemists who are primarily wet lab people say, oh, that stuff doesn't work. And what they're referring to primarily is DOC, which is a system that has been used, which is only molecular dynamics, no quantum mechanics, for many years. And it was used because QMMM was too computationally expensive. And it still is if you enumerated it. But with the cloud, you got one speed up. With the heuristic search, you got a substantial speed up. And with Moore's law, you got another speed up. So compared to just 5 years ago, this really wasn't practical. So docking is very fast, but its accuracy is about 20%. And by what that means is if I get docking scores for 5 molecules, one of them will be correct and the other 4 may not be. And so the attitude of the chemist is, okay, so I have to synthesize 5 molecules to get one good one. Not so bad compared to screening 10,000. Except, I'll tell you why this isn't good enough, even though it is 20% in a moment. A free energy calculation is where you do the Schrodinger wave equation for the binding and you enumerate instead of using the smart methods. And the problem is it takes years to do the calculation. So with inverse design, we use the AI search and the cloud. We're accurate greater than 80%, demonstrated multiple times in terms of binding prediction. And the search is we call it novel because we're searching molecular space. Typical drug discovery project using robotic screening is screening existing molecules. And so you hear stories like small molecules all the good ones are taken. You may have heard that if you talk to chemists in the drug discovery business. It's all the good ones are taken because they only look at the ones that they've already built or that they've already synthesized and they have in their refrigerators, or libraries that are variations of that. But so we have a greater novelty, greater accuracy, but let me give you an example of why this accuracy is even more important than it appears to be. In other words, the chemist's story of, oh, we only have to synthesize 5 molecules and we get a good one, isn't quite where we're at. So suppose a protein that's getting a lot of interest is JAK3 because it's indications of inflammation and rheumatoid arthritis, which are huge markets. And we're running inverse design on JAK3 now. But it's really not good enough to just have a JAK3 inhibitor with good druggable properties because there are other JAK-STAT signaling proteins like JAK1 and JAK2 whom you do not want to inhibit even though you're inhibiting JAK3. So the problem is more complicated. You have to be selective. So we were asked by a potential customer, can you give me a JAK3 inhibitor that's selective against JAK1 and JAK2? And we said, we believe you can, and we've set out and we're doing that now. But if you look at these accuracy scores, to come up with a selective JAK3, selective against JAK1 and JAK2, you have to take the 0.8, or 0.9, we'll use 0.8 to be conservative, and take it to the third power, because you have to run it against 3 protein models, not just one. So that says that 52%, or just call it 50%, of our results should be accurate. If I do it with DOC, 20% to the third power is less than 1%. So you can't design selective molecules with docking, because the selectivity will kill you because of this low accuracy figure. Yes, if all you care about is binding, and that doesn't make up a drug, just binding. You need binding, you need selectivity, you need good druggable properties, and you need low tox. And you know, we're not a tox software company, but we use tox software filters so that our output library eliminates molecules that have really bad tox scores. And there's plenty of room for improvement in that tox software, but it's getting better.
Question?
Yes?
If docking is fast, so you could have it almost for free compared to what you're doing, can you combine the two, even though docking is a less accurate score? Could you add in?
There are smart ways that you could combine the two depending on what you're trying to do. That's a good question, and I'm not sure we fully exploited that yet, but when we're interested in selectivity, because inhibitors or agonists alone aren't enough. Again, to be a molecule worthy of going through preclinical research, you need an inhibitor. It's got to be selective. It's got to be low tox. It's got to be synthesizable, and it also has to have good druggable properties. So we need to use filters to do that, and the medicinal chemists have to like it. In other words, it has good clearance properties. It's not going to go in there and clear out right away so that it isn't ineffective. Yes?
Have you looked at the correlation between the errors of docking and the errors of the new method to see if if they're completely correlated, then of course there's no extra value of using docking also. If they're completely uncorrelated, then there would be value.
That's a really good question, and personally I don't know the answer. Shahar Keenan, our chief scientist, would probably know the answer, and I will follow up and respond to that, because that's a very good point. So this is how inverse design is configured conceptually. The workhorse part is the binding calculation, which we're using pDynamo for, which is open source software. It's very good open source software. But our value added is not the computation of the Schrodinger wave equation in pDynamo. It's selecting what molecules to compute it on. So the way this all begins is we have a binding affinity equation that's applied to a target. You start with a target. The target is an X-ray structure. We build a computer model from the X-ray structure of the target, which is a protein. Step one is to calibrate the inverse design algorithm based upon any published data for any inhibitors whatsoever for that target. So this setup process is still a little bit manual. We're building automation to combination expert systems and automated algorithms to take the person completely out of the loop. This here is completely automated on Azure. This setup process takes anywhere from a couple days to a couple weeks per protein, but once it's fully automated, it'll be down to hours, if less. The library design is a process. Part of it, we're not searching complete blind molecular space because it's 10 to the 65th big, but you can choose a smart scaffold that fits the binding pockets for that protein. And we are in the process of writing an automatic library designer, but it's still today based on mining the literature. So all this setup, again, takes a couple days to a couple weeks per protein. We expect to shrink that down to hours. We've got Terry Hobbs, one of our computer scientists, working on that right now as her main mission. And as to by this time next year to have that down to hours and not days. Property filters, that's where we use third-party software to eliminate bad molecules that are bad for other properties. If they got predictively bad tox, which is not our core competency, but other people do that, then why consider it? solubility can be, or rather synthesizability can be estimated from properties, and those kinds of things go in these properties. And then we do the iterative runs where you go and use up to X molecules. We said 1,200. We were doing 500, but we're going up to 1,200. I'm not sure we got our limit raised to 1,200 yet, but we'll find out soon. You do a run on that many simultaneously. It's embarrassingly parallel. Each one is running a QMMM process, or QM process, and giving the scores back. Then it iterates. We do it again and again, several dozen times, and we come back with a new chemical entity or a small, highly focused library of several new chemical entity possibilities. And we can do this. We're doing this primarily for small molecules today, but we expect to do it also for peptides. Short peptides, not proteins, but short peptides, 10 to 20 positions. Not, you get any bigger than that, and the computation gets ridiculous because there's 20 amino acids per position to consider. Whereas if you're using small molecules, you can limit the number of R groups that you vary, and the number in each group set can be substantially less than 20. So this came from Duke University. It was validated with HDAC8, which is the protein that was done at Duke, and a new molecule was designed and synthesized, and literature data was used to validate the binding scores. And here is where we got a result of about 80%. We've also done it since it was licensed from Duke for JAK1, JAK2, and JAK3, and the correlation to the literature is good. We're getting ready to do the big production run on JAK3 that's going to, the selective one, and so we expect for that one we're going to hire out hire out a chemist to synthesize the results that we get so that we can get some wet lab correlation validating even further than what was validated at Duke. So I've already pointed this out. Benefits is accuracy, speed, and speed relative to free energy calculation, not speed relative to DOC, and novelty. And novelty, we think, is really important because, for the reason I mentioned earlier, you still hear sometimes the people in the industry saying, well, the good small molecules have been taken. Well, with space 10 to the 65th big, how can that be true? It's just that they use the same ones all the time. Hold on. Some of the business propositions that we are experimenting with in the market now are as follows. One option is, and we're an early stage company, just primarily been in development. Most of our revenue to date, in fact, all of our revenue to date has been consulting and services. Option one, think of this as a customer's option. We do a single target discovery project, which may have a total price tag of $50K or higher, depending upon the complexity, and we would ask for royalty if it ever goes to market or milestone fees, but that are much smaller than what a biotech typically asks for developing a single molecule. Option two is to license molecular libraries. In our project with Microsoft, we are doing 25 targets that we select, developing small focused libraries, a library being maybe 6 to 12 molecules big of our best results, and making those available. And if someone wants to go forth with them, they can buy them or license them or license the rights from us, or we can be collaborative with them and raise money together for the project to take it through the clinic on an outsourced basis. And this is what, in our agreement with Microsoft, has been called the speculative business, because this is where we choose the proteins in advance and work on them and then look for partners for the results. We have a partner in Philadelphia called Nemoda Technologies, who has a database and it uses SQL. The database matches molecular assets to anyone in the world who's interested in those molecular assets, either to license, to partner with, to research with, or from a market perspective. So we're using that as one of the ways that we're going to find partners for the work we're doing together, is through their database. Third option is an R&D partnership. If we find a target that's of interest to a big pharma company and we achieve early results, then we will seek a partnership with them where we do, they fund preclinical development together, or that we pass that on to them, depending upon what their preferences are. And option 4 is to license inverse design for internal use. We haven't done that yet. I think we are going to wait about 12 months till we get a bit more experience with it ourselves and make it more foolproof, and then we make that as a high-end Azure application that we'll train a company to use.
And is that an option due to pharma's concerns about, complete and utter privacy?
It could be, and if that happens, what they're going to want a private Azure. How do I say that? Azure. Both. Okay. Now I know why I'm schizophrenic with it. So that might be something that is more in Microsoft's world. What happens if a pharma company wants the cloud internally? How do you solve that problem? Do you just sell them a monster machine inside their firewall? And if so, then we can port the software over there and do a contract with them and give them a big license to do all of their proteins, or one of their proteins, or whatever they want to do. And so we're a little bit opportunistic about the business model. I think there's going to be some changing dynamics in this market, and we don't claim to have a good enough crystal ball to know what the stable business model or which one of these is going to be the driver. So we're going to spread our bets and be nimble. And as the market evolves and as this matures, it may be that we zero in on just one of these as our primary business model. But for now, we're going with the flow. And I don't think anybody in the market knows what the market's going to be. As the blockbusters move toward personalized medicine, there's going to be lots of changes. So far for marketing, we've been to bio IT world and bio, also Boston Biotech, the Boston Biotech CEO meeting I went to. We want to market inverse design to large pharma companies, these are not customers. I just put their names down as examples of the kind of clients we would like to have. We would like to partner with other Microsoft partners. I named a couple here because their software might be compatible for either improving the speed of what we're doing or being a high-end option for our customers. And we would like to market the products. When I say products, I'm speaking of molecular products that come from the speculative business using one of those business models that I mentioned, and this will be a thrust of ours going forward. We have some other bioinformatics capabilities that I would like to mention, and I also like to talk a little bit about some of the things we're interested in. PDB, which is a database of the National Institute of Health, and it's also it's worldwide. It's European Molecular Biology Institute also hosts PDB. Has been ported to it's been ported to Azure, not to the data marketplace yet, but it will be soon. One of our developers has ported it. It's in SQL Azure, so we can do full SQL queries on PDB. So we can do more powerful queries than you can do on PDB on the public site. For instance, you can find molecules that have certain kinds of properties. Maybe you want a molecule that has 3 zincs close together. You can't find that in PDB, the public PDB now, but if you have SQL, you can. And we're going to provide you with some queries that we think that are unique to the SQL Azure PDB as a follow-up.
How big is the PDB today?
About 100,000 proteins and about 300 gigabytes in that range. Our staff has both computational chemists and bioinformatics people as well as software folks. And so our reason for being interested in PDB is because we can draw the X-ray structures from that to feed into inverse design. But along those lines, we're interested in, and I had several discussions today here, we're interested in finding a natural language or search capability, semantic search capability to complement the platform, because we would like to compute upon finding adequate capital all of the targets in PDB in advance of anyone doing drug discovery on them, just so that we shorten the drug discovery cycle for small molecules and peptides at least. But not all proteins in PDB are suitable as targets. Many of them would never be a target because they're either not human or they're not part of a pathway that's of physiological relevance. So by having a literature extractor, natural language focused on biomedical literature, we can identify, which 10% of the PDB are possible. They might be targets because they were found in pathways that molecular biologists have identified in their research. So that's one literature extraction problem. We have another semantic search problem we have to do, and that is, as we produce these molecules, we need to do patent searches. And the patent searches are not as simple as text searches because we need to semantically model the properties of chemistry in those queries so that we can find whether there are structures, molecules or structures that we might be in violation of if we tried to sell these molecules, and we have to eliminate those. So we need to do, and that's not as good as a IP attorney doing it, but we're doing two-step IP. The first is this sort of automated filter, and the second one is if we have an interested customer, we have a real IP attorney look at it. And that way we're not paying attorney's fees for every molecule, but only when there's a customer and it's been filtered in advance. So we have a need for semantic searching and literature extracting in the biomedical literature, and we've already started some discussions along these lines. We're exploring a search engine, and one of your staff is also looking at what you're all doing in natural language to see if there's a fit there. And so let me summarize where our status is. The SQL Azure PDB is ported, not yet released on the data marketplace. We have some productization work to do, such as a user's manual, a privacy policy, and a license, and that stuff is being worked. And we need to do a little testing, but we'll probably have it out there for free for a little while, maybe by Labor Day. Inverse design is ported and debugged, although we found some new things that we had to do this week. And it does the heavy lifting. We need to do more front-end automation before it's released as a piece of software that we can license without hand-holding. But that's a goal. And the first the first 7 targets have been identified, 3 calibrated, JAK3 production's ready to run. We're, again, 15 full and part-time folks, quasi-virtual. We have incubator office space, but half of our people are not in North Carolina, so obviously we're not all there every day, all day. But we use the incubator as a place to go for meetings, to meet customers, and to have group meetings when needed if we're not doing it online. We're also raising a round of funding. We have an interested investor. We're looking to add to that, and we're expecting that will come to a conclusion in the next couple of months. So I have a chart that I call the Holy Grail, and I've already alluded to this, and that's the Holy Grail is we'd like to reduce drug discovery to a simple SQL lookup or search lookup. In other words, now that's a long way off, but there are significant steps toward that we can take. So we pre-compute inhibitors for all promising-looking targets in PDB. That's the immediate goal. That's an expensive computational proposition. We're doing 25 right now, and we're shrinking the time, and we're trying to do as many smart things. And we'll look at your suggestion about the combining of docking with to see if we can get any savings there. But ultimately, we're going to, you take 100,000 proteins in PDB, and we want to maybe choose 5 to 10,000 of them that we want to pre-compute this for. And you know, we said on an earlier slide, hey, well, that's a 50K engagement. Well, that includes profit and markup and people that we're cutting out. So it may really only be $10,000 worth of cloud time per protein, and that will come down as costs come down. However, it's still a $15 or $20 million proposition, so we have to raise money to do it. And our intent is to raise money for some of that, to get customers to pay for some of that, and maybe to get some of that from government sponsorship, and just over time build up enough, roll up enough money there to do this, the first step toward the Holy Grail, where we'll take the X-ray from PDB or wherever it comes from, if it's a proprietary protein. We're working on what we call the automatic scaffold designer. That has to be done before we do this in volume. That's the part that's people intense that we're automating, and the methods to automate that have been identified, so doesn't require a scientific breakthrough, it just requires more work. And what we want to do is compute this big inhibitor library, and drug discovery then becomes, you know, one of the things you'll do in drug discovery is look up and see what inhibitors are already available. And then if a customer wants them, we either sell them to them or take a royalty, or whatever business model this evolves to as part of our business. So thank you for your time. And we can do some questions if there are any. Yes?
And you had a bullet point that I don't think you spoke to, which was not just for drug discovery, for material science as well.
Oh, yes, I neglected to mention that. This inverse design technique originally came from material science in the chemistry department at Duke, and then it was, Duke got grants to do it for drug discovery, and Shahar Keenan, who's our Chief Scientific Officer, was at Duke at the time as a postdoc, and she did the original inverse design for drug discovery. But with some changes, we can use it to design materials. We haven't done that yet, and I think that what we would do initially is to do that as a service, if we find an interesting project or a customer who's interested in doing that, we can go back and optimize the materials to certain properties. What inverse design does is it maximizes the score on a property. In the case of drug design, that property is binding. But in the case of materials, there are other properties that people are interested in, and you have to change the property equation. And so there's some testing and some changing of the scope and size of the problem would have to be done, but we would be interested in branching out into material science as well, because we think it has a different risk profile from a business sense than drugs do. Drugs are, you know, low probability of success, but big money when they succeed, whereas material science would probably be a little bit more stable, so it might be good to complement two businesses, but we're small and focused at the moment, but yes, we would be interested in that. So if you know of others interested in your community in that problem, we would certainly like to have conversations. Any others?
So you've been talking more about the business side of things, but I was wondering, so in the transition to Azure from your proprietary cluster to Amazon to Azure now, has it been easy? Is it
well, we weren't really all that far. We weren't really all that far with Amazon. We had only done a couple of runs when I first came in here, and then we were given some Azure time. It was a little bit challenging for our folks at first because they didn't have, they're mostly Linux type, you know, C programmers that didn't know the Microsoft platform all that well. But we recruited this, a guy from Florida, the one who did the PDB work, who's a very strong database guy, good software engineer who has had experience with Azure, and he basically coached our staff on, getting through some of the, we got some good tips from Microsoft people too, but they had to, go through periods of time of not knowing a bunch of things until they, and so it was a lack of familiarity with the Microsoft platform as a developer, and so we brought Eric in, who's done the PDB work, and he's helped the others, Terry and Bill and Shahar, overcome the we don't know the Microsoft platform, and I think we're past that hurdle mostly.
From the platform, so other than there's the learning curve that you're coming up on, you think that the capabilities and everything you're trying to accomplish, you said embarrassingly parallel, and I tend to use the term pleasingly parallel because I'm not embarrassed by parallelism at all.
Well, I'm not either, but that's the term that the technical folks like to use for it. So I'd rather have it that way.
Yeah.
Then you cut some cost out.
But you're finding that you've got access to everything you need, or are there issues that you still have in the platform?
Well, it's taken me a while to learn who to call at Microsoft, but I've gotten more comfortable with that now. And different people at Microsoft have different ways that they respond to messaging. Some will respond to email, and some you might need a calendar appointment, or somebody might need a text. So you have to kind of figure out. Okay. So that was one challenge. That's a non-technical challenge. Getting the people trained was a challenge. And then we've run into a couple of technical barriers on Azure that your staff was very helpful with also. We're working to find the best data centers for these big jobs. We ran a job on a Friday night once, and it took a long time to queue up. Not that long. Since this is being recorded, I won't say the numbers, but I think it's gotten better since. So.
Okay, thank you.
And we're going to follow up with having some of our technical folks, especially on the PDB, talk to your SQL Azure folks. Any others?
You had suggested that your process needs some automation right now. How much, how much manual effort does it take to do one of these runs?
Well, here's the part that's manual is once we choose a protein, you have to go get the X-ray structure and set up a computing file, that doesn't take too long, but it's not fully automated. But the part that is a little more challenging, there's two of them. One is calibration, where we have to go to the literature, grab any data that's available, pull it out, and run a calibration run for that protein. And if we could do that with a literature extractor, we could largely take the person out of the loop on that. And then the other one is the scaffold design. The scaffold is largely based upon the binding pockets of the protein. We don't want to just do blind molecular space, it's too big. So we have to, are we doing a peptide or are we doing a small molecule? There's no scaffold with a peptide, we just have to decide how long it will be. But for the small molecule, you may have some parts that are going to be fixed and many parts that are going to be varied, and you have to decide how big you want that molecule to be. And the literature can give pointers to that, chemist's intuition. So this is the hardest one to automate, but we're working on it. It's going to be part expert system and part extraction and part assembly. However, those are not, the reason we're using Azure was for the QM part, which is the real number crunching where we need a lot of parallelism. This automation is more along the lines of if I want to queue up a lot of proteins, I want to get people out of the way so that I can streamline them rather than have a 2-week delay for each one or have multiple people in parallel for each one, cut that labor down. So, and also to release this as an application to customers, I think we really have to keep that much simpler than that setup is now. It's not terrible, it's just not as automated as the rest of it. We focused on the heavy lifting thing first. So it's just where we are. I don't see it, and there's some hard problems in there, but there's not impossible problems in there.
What literature are you searching? Do you just go on to archive?
Well, what literature should we search, you mean? Or do we search when we set it up? Usually PubMed. We want to know what biochemists and molecular biologists have found when they've either done binding experiments or they've done pathway analysis, depending on which problem we're looking at. The pathway is really more to determine, is this protein potentially a target? If we're doing preemptive calculations, we don't wait for a validated target. If we're doing a service for a customer, they'll bring us a validated target. So there are two different models there. And in the project we're doing with Microsoft, that's the preemptive stuff. We're not, the ones we're hand-picking initially are validated targets that are coveted by the industry. But when we get to looking at the bigger piece of the database, we have to have methods of selecting that are smarter.
Could you tell me a little bit about the calibration process that you do?
It takes a set of binding data for anything that has been bound to that protein and uses it to tweak the parameters in the algorithm. Okay, so it's an...
like a linear calibration where, you know,
No, but it's...
a 0.6 that you predict gets mapped to a 0.75.
No, but there is a program that does it. You just have to find the inputs that you want to use.
Gotcha.
And if you want the detailed science of that, I'd have to set up a call between you and Shahar so she could share, you know, what's in the algorithm. But the algorithm exists. It's a matter of finding the data that we're going to put in it. And the same would be true if we did material science, for instance. We'd have to seed it with anything that was known. Now, it's possible to do this without calibrating, but it takes a two-step process. In other words, you find a homologous protein and get some of that data, run with that, take the results, then you have to synthesize it, get the binding data, and run it in again.
Right.
Or unless you get really lucky and it's good enough, then you don't have to run it again.
If you're looking for binding to a single site, does calibration affect the sort that you would do afterwards of what binds strongest to what binds weakest? Or is it only when you're talking about, you know, you want something that matches this and doesn't match these, that now you really need calibration because you have to be able to combine?
No, that's selectivity. The selectivity is done by running the result set against other protein models. All the proteins have to be calibrated. Others?
So I want to thank you very much.
Thank you for the opportunity.